Welcome to APFCB webinar on role of clinical laboratories in COVID-19. In this lecture, Professor Khosrow Adeli will be talking about IFCC guidelines on molecular and serological testing of SARS-CoV-2 and biochemical monitoring of COVID-19 patients. Dr. Adeli is currently the head and full professor of clinical biochemistry at the Hospital for Sick Children and the Department of Laboratory Medicine and Pathobiology at the University of Toronto in Canada. He currently serves as the president of the International Federation of Clinical Chemistry and Laboratory Medicine, IFCC.
I want to start by thanking the Asia Pacific Federation of Clinical Chemistry and Laboratory Medicine for the kind invitation to give this presentation on the IFCC COVID-19 guidelines. As you may know, IFCC formed a task force on COVID-19 back in March at the initial stages of the crisis. And this task force with IFCC support, has been examining all of the evidence in the literature and has been holding many discussions and has been very active developing a number of guidelines. And I'm happy to present some of these guidelines today. The official publication will appear later this month in September. So you are actually one of the first groups to hear about some of the new IFCC guidelines in this area. This is a presentation on behalf of the members of the IFCC task force on COVID-19, which includes a number of international colleagues from around the world, as well as industry representatives and regional federation representatives across the globe. Now, as you are well aware, the clinical lab plays a critical role in any infectious disease monitoring, and of course, in this case, in COVID-19, has been playing a crucial role in managing this pandemic through clinical testing. And this includes diagnostic testing, serological monitoring, as well as when patients are actually hospitalized, they are then tested extensively through biochemical tests, as well as hematological tests to assess their metabolic health and progression and perhaps severity of the condition can be determined through these testing. So lab medicine clearly plays a role from the initial diagnosis until a patient is monitored, treated, and released from the hospital. So it is a critical, really part of the team that first diagnoses and then monitors and treats the condition. So as you know, there have been many different tests developed, including molecular tests for diagnosis, serological tests for detecting the presence of or exposure to the virus, as well as I mentioned, when patients are actually hospitalized, there are many other tests that are used to assess these patients, particularly biochemical and hematological. So I'll be discussing these tests and then focusing on the guidelines that IFCC have put together for clinical labs, particularly clinical labs in the developing countries where guidelines could be particularly helpful. There, as you may well be aware, there have been many controversies around some of the testing in terms of quality offered by many different manufacturers around the world. And so ensuring that you have the appropriate test and you validate that appropriate test is critical, of course, in ensuring appropriate test utilization. Now, this slide nicely, it's one of the recent publications showing the different stages of testing. Now, as you know, the molecular tests are used initially to diagnose the presence of the virus. And this is usually a fairly limited period of time where the testing, the virus can be detected. Many patients, as you know, and many subjects are infected and are do not have any symptoms, and therefore the virus can be present and lost fairly quickly, usually within a few weeks. But in some cases, it may persist longer. But typically, the period of detection is relatively limited. Now, soon after the infection, there is this immune response, and therefore serological testing can detect the presence of either IgM, IgG, as well as IgA, which is not shown here. But typically there is a response and therefore development of antibodies that can be detected. And as I mentioned, for those who are infected and then develop significant number of signs and symptoms, they are then hospitalized, and there are a large number of biochemical and hematological tests to assess health at different levels, including cardiac health, kidney health, liver health, and systemic health. And so therefore, a number of my biomarkers have been utilized accordingly. So we have developed, the IFCC task force have developed guidelines around molecular tests, serological tests, as well as these tests used in monitoring patients. The next slide just shows you the IFCC website. We have actually, one of the first things that was done back in March of this year is we developed an IFCC information guide. It is on just this week on September 1st. But this guide has been updated since March on a weekly or bi-weekly basis. And I highly, those who are not aware, I highly recommend visiting this guide, because the guide has a very extensive literature cited, some of the more higher quality publications are cited and discussed, and there is information on many different aspects, not just testing, but many different aspects of COVID-19 crisis. Now, in addition to the website, as I mentioned, a task force was formed, and this task force, the terms of reference, is actually not only to establish that platform, which I just showed you, the guide, but also to look at guidance, to develop guidance and guideline documents. And those are things that I will discuss today on diagnostic and serological tests, as well as others. But we also, this task force, headed by Professor Giuseppe Lippi from Italy, actually developed initially, the first guideline that was published was actually on biosafety, laboratory biosafety. And laboratory biosafety publication has already appeared in CCLM, and those who haven't seen it, I recommend downloading that free publication. Because it can really assist in labs biosafety, focus on managing biosafety within the clinical lab. But there are a number of other terms, because for sake of time, I won't discuss, but this task force is one of the key sort of terms of reference is actually guidance documents, and that's what I'm talking about, guidelines on testing. Now, these are actually some of the publications from the task force. The initial biosafety measures that I discussed already appeared in CCLM back in May. The two surveys actually developed largely by Dr. Tessa Bing from Singapore and working with the task force. And both have now been published in CCLM, and a critical review on testing that was published also in CCLM. This was published in a series of articles from IFCC. And again, if you haven't seen these, they're all available for free download on both IFCC website as well as on CCLM journal website. Now, as I already talked about the task force terms of reference, but this just further highlights that the most recent focus has been critically reviewing the published literature and then developing guidelines. And there is, as I mentioned, a very large number of people involved, and these guidelines will have an international sort of support group that authors these guidelines, and it includes medical clinical biochemists, microbiologists, virologists, as well as scientists representing the diagnostic industry. So it has wide representation. And the guidelines are, there is actually 3 documents that will be published soon. One on molecular testing, one guideline document on serological testing, and the third is on biochemical hematological testing. Now, let me start on the molecular testing guidelines. And these are basically particularly important in diagnosis of disease. And as you know, molecular diagnosis usually involves a nucleic acid amplification test or NAT tests. And these are meant to detect the virus and the viral RNA. Since this is a RNA virus, one needs to target the RNA for detection. And this is a map of the viral genome, the COVID-19 virus, also called SARS-CoV-2, which is actually the actual name of the virus. And this has a number of regions that can be targeted. And so different companies and different assays have different target regions. So they measure specific targets within this virus. And a number of methods are used to amplify and then measure the presence of the virus. So it's really a PCR-based assay or nucleic acid amplification assay. So in one way, it's a pretty straightforward assay, but the complexity comes from the target regions, the design of the primers, as well as the detection technology that's used. Now, when would you when would you do molecular testing? So based on these are IFCC guidelines that indicate that the obvious indications, of course, is to diagnose the viral infection, especially during the acute phase of symptomatic illness, when there is actually symptoms present. And then to assess or to assist in clinical assessment of those who do not have any symptoms or are pre-symptomatic or mildly have mild symptoms with known exposure to positive cases, and then to assist in screening during various conditions or in various populations. For example, prior to a scheduled surgery, right now many countries are facing the challenge of opening schools or going back to work. So the testing, of course, is helpful in those scenarios, and managing small outbreaks. And there have been many of those, as you know, in many countries. And then populations also should be, based on these guidelines, should be prioritized. Certain populations should be prioritized for molecular testing, and those are listed here, which are fairly obvious. For example, those with acute respiratory illness, and those who are known to have been in contact with known positive cases. And then high-risk groups, such as the elderly and patients with, for example, immune disorders or other pre-existing conditions. For sake of time, I'm going to go quickly because there's a lot of material to cover. In terms of assay selection, there are a number of guidelines. These are just summary. The documents provide much more detail. So I suggest that you visit those guideline documents, download them when they are available, probably within a couple of weeks. The clinical, in terms of selection for molecular tests, labs should select an appropriate NAT or nucleic acid amplification test, and then ensure that it has appropriate validated performance. And in many cases, point of care assays have been found, at least the initial ones that were developed were not, the evidence did not support their use because of performance issues. But I have to say some of the more recent point of care assays, rapid tests, have had much better performance. So of course the technology is improving very rapidly. Also, there is currently insufficient evidence to suggest the definitive advantage of using a specific target. So many different companies and kits and even homemade assays target different gene regions. And it's not clear whether one is preferred over the other. And then usually assays should be actually targeting a minimum of 2 regions or gene targets. In terms of the specimens, there's a number of issues, and I don't have time to go through the details, but clearly one has to be aware of what kind of sample. At the minimum, an upper respiratory tract specimen should be collected, and samples such as saliva have been used, but the evidence is not as strong supporting their use. And a number of other details that are in the guidelines. Next, in terms of verification of regulatory approval, one, of course, must performance claims. One should not just bring a kit in and start using it. It should verify the manufacturer specifications or claims and performance claims, and then be involved. The lab performing the test should be part of a quality assurance program. And now there are QA programs available for COVID-19 molecular testing. Also for serological testing, some are starting to provide service and materials are available. Labs should verify clinical, not just analytical, but also clinical performance claims in terms of sensitivity, for example, and the specificity in the specific populations. And then, of course, samples matter, and one should ideally use clinical samples, for example, known positives and non-control negatives. But if unavailable, then there are these contrived samples available that can be used in place of clinical samples. And there are guidelines available. If one develops a test and tries to publish it, the guidelines that there are certain guidelines that dictate how that performance should be evaluated and reported. And so START guidelines are critical, especially if one is trying to report a new assay performance. Now, there is a lot more detail, but this slide simply lists some of the key parameters that need to be verified, such as limit of detection, reportable range, imprecision, accuracy or trueness, as well as analytical specificity. So these are some of the key parameters, and there's a lot of information. Again, I recommend visiting the guidelines when published very soon. Now, in terms of reporting and interpretation, there is usually, of course, either a positive result reported, negative result reported, or in some cases, it's indeterminate. And the positive result usually means that it's been confirmed that the viral RNA is actually present, but you cannot judge based on a positive result. You cannot judge the presence of active viral replication and potential for viral transmission. So because there is now actually more recently evidence that the level of RNA, using quantitative assays, one can determine what level is present, and that might assess or predict or the potential for transmission, because low viral load may actually mean that the risk of transmission is low. And so therefore, a positive result, which is usually based on a qualitative assay, cannot predict any of these other sort of parameters. But certainly it confirms the presence of the virus. And also, by the way, one cannot predict presence of any disease with a positive result, because many patients, as you know, are asymptomatic and cannot necessarily show much symptoms, or do not show much symptoms. And therefore, a positive result is not predictive of illness. A negative result is usually reassuring for subjects or patients, but again, there are cases of false negative results, and therefore potentially retesting may be necessary depending on signs and symptoms of that subject being tested. And then there are cases where results cannot be fully clearly interpreted, in which case repeat testing, usually follow-up testing, retesting is recommended. Finally, I mentioned the limitations, and there's actually a large number of limitations of these assays, both pre-analytical and analytical. And based on the IFCC guidelines, one should be aware of the main limitations, and that is the possibility of false negative results. And that could be largely due to pre-analytical errors or analytical errors. Actually, in many cases, more pre-analytical than analytical, and false positive results, and delayed timing of testing is another contributor to sort of either indeterminate or false reporting of results. So there's of course issues of resources and testing capability of the lab, especially in developing countries. So these could be factors in quality of the reporting. So I'm going to move on to serological testing. Serological testing is becoming very common. There are many kits available on the market in North America, Europe, Asia, everywhere, and there are quite a few of them developed, and not all of them very well validated or approved by FDA or similar organizations. Here in Canada, we have the Health Canada approvals, which is similar to FDA approvals. And only certain tests have been approved. Many of them have not. So one has to be aware of which ones are actually approved for use by reliable organizations such as FDA. And there are similar organizations around the world. Now, serological testing come in different flavors. There's some rapid diagnostic tests. Well, they're called diagnostic, but really, in many ways, they're not diagnostic. They are just simply assessing presence of or exposure to the virus. It does not necessarily diagnose an infection, so that has to be clear. Diagnosis is not perhaps the most appropriate name for a serological test. It's really assessing exposure to the virus. But the term is used quite commonly in the literature. ELISA-based assays as well as neutralization assays have been developed, but the most common ones that are used in the lab are ELISA-based assays. But these are automated ELISA type-based assays using different detection technologies such as chemiluminescence or others. Neutralization assays are actually quite helpful, but they tend to be more difficult to perform. They need cell culture techniques, and therefore they are limited. They're only present in certain reference labs currently and not widely available. But ELISA-based assays or automated ELISAs are most common. Now, again, in terms of indications, the guidelines have laid out clearly what the indications are. Some of them are very obvious, but the key thing is that one thing that should be remembered is the take-home message. A serological testing, again, is not diagnostic of presence of the virus. It's more assessing exposure to the virus. And so therefore, the indication, clear indication is one is to serve as an adjunct to molecular testing in patients presenting with suggestive clinical features. This actually happened in our own hospital, in a children's hospital, where we have many people in the last few months, children coming into the hospital who are actually PCR negative. So they do not have the virus, but they do seem to have signs and symptoms of COVID-19, which suggests that they have had exposure to the virus, because then when we perform serological testing, they're actually positive for serology positive, suggesting that they have come in contact with the virus, but then develop signs and symptoms later. The virus has disappeared, but there is an immune reaction that can be detected. And that immune detection actually can seems to lead to certain complications, clinical complications. There are a number of other indications. The one that I just mentioned is actually pediatric related, called MIS-C, which is a multi-system inflammatory syndrome. It's fairly, it's not very common, but it's not uncommon to see that in our own hospital. We have seen quite a few cases where I work in the children's hospital in Toronto, and this has been seen quite a few times, and we still have quite a few children with this condition. Some of them have actually been hospitalized and have had severe symptoms and complications. But many of them tend to have mild conditions and are sent home and monitored. Again, there are many potential applications, such as seroprevalence surveys of the population and many others. But again, I don't have time to go through every one of these indications, but there are quite a few possible indications that can be helpful using these assays. And these assays, again, measuring either IgG, IgM, or IgA, most commonly IgG. And there are certain indications that are not supported by evidence, and that's been listed for example, to diagnose the infection in the acute phase of the disease. This is, as I've already mentioned, a real diagnostic test, the serology test. Now, in terms of assay selection, there are quite a few options available. There's, as I mentioned, different types of immunoglobulin that can be measured, but also there are largely qualitative assays, but more and more quantitative assays coming on the market. There's also assays based on different measurement technology, ELISA, ELFIA, as well as CLIA assays. And these seem to have different types of sensitivity and specificity. And I've already mentioned that there are a wide variation in the quality of these assays, these immunoglobulin assays, and one has to be extremely careful how to select and verify the various parameters, quality parameters. Now, as I mentioned, neutralization assays are also available, but they're not really not really found in or useful for routine labs because of the complexity of using cell culture, and so they are relatively uncommon for routine use. They are used more to confirm. For example, if you find a positive serology assay in a patient, and we have done this in our own studies, where you find certain positive serology tests, you can actually confirm it, try to confirm it using neutralization assays. And so that's a different, so that it's more of a follow-up assay. I've already mentioned the point of care assays. Point of care assays are becoming more and more available, but the quality is thought not to be the same as lab-based assays, so there's still a lot of controversy in this area. More details in the guidelines can be found in the guidelines. In terms of verification, there are a number of parameters that need to be verified. Again, looking at analytical performance, looking at clinical performance, and again, ideally one, if possible, to try to use a quality control program and participate. And there is actually now CAP surveys available, as well as there are surveys available through other quality assurance companies and manufacturers. And again, when developing new assays and trying to publish those parameters, one has to follow the START guidelines. Details to be found in the guideline documents soon. Now, in terms of verification parameters, again, similar to what we saw with the molecular test, there are a number of parameters such as in precision, carryover. Carryover, by the way, is an important parameter. We have seen it in our own lab. One has to be careful. For example, if you run positive samples after you run patient samples, you could potentially have carryover and therefore have false positives. So one has to be very careful how you actually run your samples. Cross-reactivity is a potential issue because there are some, there is evidence that the antibodies against some other coronaviruses could recognize, for example, could interfere, I should say, with assays for COVID-19 serology. And then interferences from other antibodies and other factors. In terms of reporting, again, you either report a positive result, negative result, or intermediate result. And it's clear that when you have a positive result, you're just really saying that the antibody is present. You cannot really determine when that virus was present, the sort of the timing of exposure, or even the severity of any condition that may be present in the patient. Because these are qualitative assays, there is also no link to clinical contacts in the sense that there is really, it's hard to use a serology assay to assess whether the patient will likely have more or less symptoms or complications. So these are really ways of assessing the population. It can be useful in seroprevalence studies to assess how many, for example, a student in the school have been exposed to the virus. Well, so that can be useful, but beyond that, the sort of indications are limited. Negative results clearly suggest that the patient hasn't come in contact with the virus, at least recently. Unfortunately, there is evidence that there are cases where antibodies can actually gradually be lost or disappear. So there is a chance that even when antibodies cannot be detected, so a negative serology test does not assure that there's been never exposure to the virus, in the sense that there may have been exposure months earlier, but the antibodies are no longer detectable. So that is therefore follow-up testing has been recommended in some cases, against if there is, especially if there is a concern. And then there are cases where it requires retesting or follow-up testing because the results cannot be interpreted well. Because, for example, they may just be close to the detection limit of the assay or other complicating factors. Now, the current limit of assays, we sort of have addressed some of these issues. Particularly important, I would say, for serology testing is patient selection or population selection, because it's been shown that prevalence of the infection in the population can be a major determinant of the sensitivity of the assays that are used. And so therefore, one needs to ensure that the appropriate patient population is being screened with these, using these serological assays. And very few studies have examined assay performance in asymptomatic patients, patients who have no signs or symptoms of the viral infection. And because the reason is that the sensitivities of these assays tend to be fairly low in those types of settings. But if you use, for example, a population that is known to have a large number of people with the viral infection, the sensitivity tends to be better. Well, because of course, probably a higher amount of number of subjects, but also higher viral load and therefore higher immune response, and therefore easier to detect the presence of the antibody. And a number of other parameters. Again, the details are in the guidelines. For sake of time, I'm going to move on. Now, the last part of the guidelines discusses biochemical and hematological markers. These are assays that actually most labs, biochemistry labs, hematology labs, are quite familiar with, but they have not been applied to more severe cases of COVID-19 patients. And this is a slide coming from, we actually published recently a review on pathophysiology of COVID-19 infection, looking at pathophysiology of the disease and how different organs are affected. And so this slide comes from that publication. Again, I believe is free to download for those who are interested. There are a number of complications that occur during infection. And of course, it varies from patient to patient, but there's a number of first physiological host responses, but also pathogenic responses that can lead to significant disease. And many assays have been used to assess both physiological response, but also pathological responses. For example, cytokines, and we actually in our lab have established 8 different cytokine tests that we have used in children who have had signs of multi-system inflammatory disease or syndrome, and that has been very useful in assessing the degree of the immune reaction, because these are very important markers, interleukins, as well as things like TNF-alpha and others, so have been very useful in assessing those patients. But there is also a number of markers that measure organ failure or damage to the heart, to the liver, to the kidney, and other parameters. And more of those are covered in these slides. So there is, depending on, of course, severity of the disease, But as you know, there have been quite a few millions of patients who have had very severe disease, and many of them have actually died, and they're because of the very severe multi-organ damage that occurs. And so this slide tries to show you some of the key markers that have been used to assess various complications, brain complications, heart complication, kidney, liver, gastrointestinal, as well as blood parameters and overall or systemic disease. And these are markers that are very familiar to you, such as many of them have been used for other conditions. But interestingly, there are quite a few papers showing how useful cardiac troponins have been in assessing these patients, and NT-proBNP for cardiac failure, and also markers of kidney disease, markers of liver health and liver disease have been quite commonly used, and there's actually many of these patients have very high ALT. Interestingly, it goes up together with high ferritin. And ferritin is not just a liver marker, but also an important marker of inflammation. And there seems to be a significant rise. By the way, CRP is not mentioned here, but CRP levels are also significantly elevated in these patients. Now, in terms of recommendation, the guidelines recommend the following hematological tests to be performed in every patient. And of course, the CBC, D-dimer has been particularly useful actually in this population and used heavily. Platelets seem to be a significant loss of platelets in this condition that should be monitored. PTA, PTT, and fibrinogen are typically measured to assess these patients. In terms of biochemistry tests, I already showed you some of the ones that have been measured, but these are the ones that the guidelines recommend. Of course, blood gases to and RP, ferritin, I already mentioned. ESR, procalcitonin actually has been useful. Procalcitonin has been useful because it's a marker of bacterial infection, as you know, and sepsis. These patients who become quite severely ill with the coronavirus also then have complications in terms of additional bacterial infections that can contribute to more severe illness and even death. Therefore, measuring procalcitonin has been useful in these conditions. Cardiac troponins, I already mentioned, ALT, bilirubin in terms of liver health, albumin, commonly measured. So some of the common assays, you're all very familiar. But I mentioned cytokines. Cytokines, again, have been very useful in this condition, and which is the reason we developed these assays in our own lab back in actually March. We had already planned to develop them for other conditions, but they have been very useful in the COVID-19 crisis. Now, there is also evidence that you can use risk scores using the results from a number of a battery or a panel of biochemical hematological tests. You can actually predict the progression of the disease or the severity of the disease and use it as a sort of a prognostic marker. So these are called risk scores. And there are a number of publications that have come out talking about risk score calculation for COVID-19 based on measuring a battery of tests. These are actually quite useful to assess which patients will likely require more care and more attention. Also, there are a number of issues or limitations that have to be kept in mind. For example, no one test should be considered in isolation. Therefore, looking at batteries of tests, which I've already mentioned, biological and analytical variation. Also, the guidelines caution that when translating a study findings, some of the recent studies, to local lab practice, one has to be aware of diagnostic cutoffs that have been recommended. Finally, in conclusion, a large number of, as you know, populations around the world have been exposed to this virus. Unfortunately, I think the most recent number was close to 26 million infections that have been detected. Remember, those are only people that have been tested. Likely the number is much higher than 26 million. So it's a very major crisis. Of course, it's really been unprecedented, and it's not been never, I don't think we have ever seen, even in looking at the history of humans, this is really a very unfortunate crisis that is affecting every corner of the world, which is really unfortunate. But there is no question that the only solution is to fight this major crisis through not just testing, but of course appropriate medical care and monitoring the population. So what I'm trying to say is that the clinical lab is a major player in this fight against the pandemic around the world through patient screening, diagnosis, monitoring, treatment, and even later looking at prevalence and surveillance of the population. This is the role of the lab is now very clear in view of not just the people working in the field, but also the public, the governments and hospital administrators. It's actually a very good example of where the lab makes a huge difference in care, in both diagnosis, but also care of patients. So I actually recommend this as a way of publicizing the role of the lab in your own institution or in your own city, in your own organizations. And I should mention a number of new, particularly interesting is antigen testing. Antigen testing has this potential that could replace potentially molecular tests that tend to take longer to perform. Antigen testing, particularly if performed at point of care, can be very rapid and can be very useful in detecting the presence of the virus, just like we do with hepatitis screening. So antigen testing is actually very useful, and it's coming, and actually there's some kits already available. So the IFCC task force has worked hard to develop a number of guidelines, and these guidelines will appear shortly on the IFCC website, but also in a peer-reviewed literature, and it will be published in a journal. It's just been submitted, and the task force is hoping to continue this work. These are, by the way, interim guidelines because the evidence is rapidly evolving. The work, as you know, many publications coming out every day, and therefore the evidence is further developing, and therefore these guidelines may need to be updated in a few months' time. But these guidelines at least would be helpful in the interim, hopefully to many labs around the world. So thank you for your attention. I hope that these guidelines will be useful to your lab.